I'll start.
Go, Alan.
When you brought up the idea or paradigm of an engineering perspective on how cells are designed to accomplish the balanced goals of reliability in the face of complexity. Another aspect of cell magic is redundancy, and how it's amazing that cells can get along with what they need to do with this gene or that gene knocked out, and there's a small list of genes that in fact will be completely lethal, but a lot of genes can be, sort of optional. And I don't know, does that come into your modeling or perspective on analyzing biology?
Yeah, certainly. That's one of the aspects, or that's thought to be one of the huge factors for reliability, having this redundancy actually. Yeah, so that's certainly an important factor. What also comes into play, I think, when you think about engineering versus how cells are made is the evolutionary history. So I think an interesting question is how much of it is just a byproduct of like how cells evolved and how much of it is really needed. And I think synthetic biology will actually be nice to address that, because you can try to kind of make it simpler. You can try to apply the human engineering approach to cellular function and see how well it works. And that will partly answer whether that whatever we see in terms of complexity is really needed or whether it's a byproduct.
So I'm viewing this meeting really as a way for us to all collaborate and learn more about each other's work. So a question that I wanted to ask all of you, and I think Silke said this, is viewing these systems as integrated networks. Can you elaborate a little bit more of what sort of network techniques you've used to do structure analysis or the dynamics on these networks?
I can get started. So as I said, the question that we have is how proteins signal from cell surfaces. And we use a wide range of biophysics technologies that involve not only studies at the structural level, but also the functional level. And we go beyond that point. Also, because sometimes when you do in vitro experiments, you need actually to demonstrate that these findings can be reproduced in a physiological condition. So we use other technologies. I haven't shown those technologies in my presentation, but we also use studies in cell-based on organismal levels. And also, we collaborate with other groups on campus and outside of campus regarding how these interactions take place. And we do also bioinformatic analysis by molecular dynamic simulations to predict not only how the interactions take place, but also how to do modifications of those interactions and predict how these interactions may occur. And then based on these findings, that accelerates our research work, and then we can do those changes at the in vitro level.
Yeah. Well, so in terms of our research, there are two areas that, because I was fooling around, I didn't really have the time to explore. But I think the connection between all these molecules. I mean, let me put it this way. The circadian molecules recognize and control the expression of genes based on recognition of specific elements, the regulatory region of genes that are very promiscuous in the genome. So they basically control the expression of very many genes. They also regulate interaction with other proteins. Now, the problem that we have in my lab is that we can check one thing at a time. So that is very minusculous, that is very reductionist, and that is a tremendous failure that molecular biology had in the last 20 years by trying to understand big processes by looking at what happened with one single gene and one single mutation. So network analysis, genomic analysis is not something fancy, something that we need these days to fully understand what's going on in a cell and the cell in the context of other cells in much larger environment. So we welcome all this analysis that can shed light on what's going on in these cellular processes.
I think we talked about this a little already today. It's a lot about the friction between biology and models, actually, how we can really fit them best. We have constant concerns about, how large a model should we take? How many components do we want to have in there? Do we just want a toy model, or do we want something mechanistic? Then the mechanistic thing or statistical models come with a completely different set of problems. So we try to match it with the question that we have, but it's actually really... It's something that needs permanent working on. It's actually almost a research in itself. Like how do you optimally design your?
Yeah, I think biologists have been very arrogant in the past by considering that large data analysis was a fishing expedition. I think we pay for that arrogancy by ignoring a lot of data that was published before that really will help us to build new hypotheses based on that data. So hopefully our generation will prove the previous one wrong, as usually happens. Power.
I guess it's one for Carla. There was a major paper in Science by Johns Hopkins group claiming that, I'm not trying to play down the significance of environmental factors in cancer, but the conclusion of the paper was that the majority of cancers are bad luck.
I read the paper.
So I'm just curious of your opinion on this.
Yeah, I remember reading the paper. Actually, I passed it to everybody in my group immediately it was published. Well, what can I say? It was a good statistical study. It has been criticized, of course, by the scientific community. Is based on the sequence of very many genomes, and there is always a component of bad luck on getting cancer. There is a genetic predisposition for some cancers. There are some driver mutations that are critical for cancer. There are 14 of them that if they are there, you have higher propensity, and I think that's what they meant. I don't think they disregard other factors. I mean, I think the scientific community took it in a very, bad way. I mean, it's like a defensive way. And maybe because the title was, a little bit, I mean, it was sexy, but, but maybe a little bit challenging. But there is a component.
Okay, with that, if we can thank the VBI Biology Fellows for some great presentations.